Good morning, everyone. My name is Mathys, and today I'm going to talk to you about discovery of transcription initiation at microstellites, which is also predictable by sequence-based deep neural network. So to begin with, I'm going to give you an introduction on microstellites, which are also named short-term repeats. So short-term repeats are sequences composed of repetition of simple elements from 1 to 6 base pairs. We are going to call the str class the element that is repeated. So in this case T, in this case AC. We are going to call the str length the number of repetitions. And we are going to call flanking sequences the sequences which are upstream and downstream of the str. So for this study, we used a catalog published by Williams et al. That repertoriates the STRs genome-wide. So knowing that, I'm going to give you the beginning of the story, which is the publication of a mammalian expression atlas by Phantom in 2014. So this catalog was built using the cage analysis of gene expression technology for cage. So how does it work? It is a sequencing of the 5' end of the capped RNA. So the reads are about 27 nucleotides long, and they are then mapped onto the genome. This is important because it means that you are going to have a tag and a location. Well, the cage method was applied genome-wide on roughly 1,000 experiments in human across multiple tissues. It has two key strengths. That is, it is precise and strand specific. By precise, I mean base pair resolution. So when we probe that catalog, we noticed that around 72% of the cage peaks detected by Phantom, as defined by them, are unassigned to promoters and enhancers. And also a significant fraction of all of these cage peaks, those DSSs, well, they initiate at STRs. And we see here, right, a browser view of the signal that we have, some cage peaks, and how the strand specificity and the precision of the cage technology. So when we try to look a little bit deeper into those STRs that had cage peaks, we got to the following conclusion. So here you have a figure. On the left, you have the number of STRs in the catalog, and we see an overrepresentation right on T and A. And on the right, you are going to have the fraction of STRs that contain a cage peak. And what we see is that these distributions do not match. And this means that the peak distribution is not random over STR classes. Otherwise, these classes would have very little cage peaks. Well, this is interesting. We try to dig a little bit deeper by developing a continuous metric, because a cage peak is a discrete metric. You either have a cage peak or you don't have one. So what we did is that we took the cage signal along the STR, we averaged it over all of the libraries, and then we divided it by the STR length. And we call that normalized type count. So here you have a representation of the distributions of normalized type counts for each of the STR classes. And we kind of see the same conclusions. You have the most represented classes T and A that are not necessarily the most expressed. And something that's also interesting to note is that you have a good Pearson correlation, about 0.87, between human and mouse orthodox. So that transcription might also be conserved between mouse and human, which is something that we discussed more extensively in the paper. Well, while this is good, it is still not enough to be able to affirmate that we have transcription at microsatellites. because of a mapping problem. If you remember well, the heliscope technology used by Cage, in the Cage technology, only sequences 27 nucleotides. And it is super difficult to map 27 nucleotides of repeats onto the genome. So what the Phantom consortium did was developing a method using both cap trapping and mean-ion sequencing, also called CTR-seq. That method, instead of sequencing just 27 nucleotides, sequences 500 nucleotides. So the mapping is much better in terms of location precision. So we then compared the result that we obtained with heliscope and mean-ion sequencing, and what we saw is that we draw the same conclusions. Still, the classes that are most represented don't necessarily have the more amount of Cage peaks, and also the distributions are very similar, right? You have a Pearson coefficient of 0.88. The only difference is the scale, right? The Y scale, and this is due to the depth of mean-ion sequencing being much less than the depth of heliscope sequencing. So this, at the moment, we had validated transcription initiation at short tandem repeats, right? So we had a signal, a continuous signal that was a trustworthy metric of expression. And what we did with that is that we tried to develop a model that could predict that expression from the sequence alone. That would tell us if that expression is driven by sequence-based instructions or something else. So to do that, we used a method, a very classic method that most of you, I think, know, is a convolution neural network, or CNN. And we took basically 101 base pair long sequences, so 50 downstream and 50 base pairs upstream of the STR from the STR end, right? So those are rather short sequences, and we encoded them into this one-hot encoded matrix, and we just pass those matrices into our CNN, which is composed of 3 convolution layers of very short filters to accommodate for the length of the sequence, and also 2 dense layers. So the model can be put into 2 modes. You're going to have a regression mode that we use to predict the normalized tag count that I presented to you earlier, or you have also a classification mode which is used to predict a wide array of tasks. So how do we do? Well, first I'm going to present you the training method that we used to prevent overfitting. So we split our data into 3 parts for each of the classes. So a training set, a validation set, and a test set. The training set trains the model that is validated continuously on the validation set, and once we see that the validation set, the performance of the model on the validation data starts to drop, we then stop training because at that point we would overfit, and then we finally test our trained model on the test set to measure performance using Spearman correlation between the observed normalized tag count and the predicted normalized tag count by the model. And what we see is that the model accurately predicts a certain amount of classes. So this validates our hypothesis that some of the transcription can be explained by sequence-based instructions. So after that, we try to dig a little bit deeper again by looking at the flanking sequences. And this is because typically in the short tandem repeats or the microstellites community, we look at the STR length and only at the STR length. And by replacing the STR, no matter its length, by 9 ends, so which doesn't have an effect onto the model, we deliberately choose to hide the length and the nature of that STR and only take into account both of the sequences on both sides, the flanking sequences. So then we train the model based on these modified sequences, and then we test using the method previously shown. And the output is going to be a Spearman correlation between true tag and predicted tag for each class. And what we see is that the accuracies of the model built only on the STR's flanking sequences tend to be still good based on the one compared to the model that use the whole sequence. So that's an important finding, is that, well, maybe in the STR community we should take into account more the whole picture, right? The surrounding of the STR to make our analysis. Something else that we did with those flanking sequences is that we tried to assess the viability of those flanking sequences between STR classes. So for that, what we did is that we took sequences from two different classes, so in this case T and A, and again we processed those sequences by hiding the STR and also hiding few bases downstream the STR to escape problems due to mismatch or misalignment of STRs within the catalog. That could help the model to determine which classes it will look at, right? We took those process sequence and again we trained the model, in this case in a classification mode, and we trained the model to the task try to find the different classes, right? So if I give you a TSTR, is the model going to be able to tell it is a T or not? And we did that for each of the classes. So the output is going to be an area under curve for each pair of classes. So in the results, we find that we are going to have a model for each pair of classes that is fairly accurate at discerning classes based only on the flanking sequence. So example here, right, our worst performing model is going to have trouble comparing two very similar types of STRs, but in other cases you mostly have a Spearman correlation, or no, sorry, area under curve over 0.7. So what that tells us is that STRs have different nucleotide compositions in their flanking sequence, and that's something that was already observed for the AC STR in 2004, and this study extends this observation to all of the STRs. So to recap, we have shown that there exists transcription initiation at short tandem repeats. This transcription is partially predictable by a CNN using the sequence as the input. And the STR flanking sequences, they contain important predictive features, and these features, they change based on the classes. So taking this information, we try to look at how these STRs might change in the clinic, right, for some pathologies. And what we saw is that the STRs that are associated with clinically important variants taken from the ClinVar database, they tend to be more expressed than the one without ClinVar variants. And within those STRs that have ClinVar variants, the ones that tend to be the most expressed are the ones that harbor pathogenic variants. So that raises a question, as we have known for some time that STRs have regulatory functions. And it could be that the transcription initiation at STRs could impact or drive these functions. And as different classes have different motifs or features that leads to different levels of expressions, maybe also different classes could serve different functions. So as a team right now, we are heading that direction and probing for finding regulatory functions correlations with expression. So thank you for listening to my talk. I would like to thank our team and collaborators and also the organizers for inviting me to present this talk here. Thank you very much, and I'm happy to take questions.